Hello students, in today's presentation we will discuss about germplasm conservation and cryopreservation. At the end of this session, the learner will able to discuss principle of germplasm conservation, different methodologies under germplasm conservation, cryopreservation principle and methodology of cryopreservation. Application of germplasm conservation and application of cryopreservation. So let's start with germplasm conservation methodology. First, we will discuss what is germplasm. Germplasm is nothing but hereditary material which is transmitted to offspring through the germ cells. The objective of germplasm conservation is conservation to preserve the genetic diversity. There are two approaches as you can see in this slide, in situ conservation and ex situ conservation. Let's discuss about total methodology come under germplasm conservation. The conservation can be carried out with the help of in vivo methodology and in vitro methodology. In vivo, that means with the support of live animals, and ex vivo, that means with the support of cryoconservative materials like oocyte, semen, embryo and somatic cells. In a case of in vivo methodology, there are again two methods, in situ and ex situ. Let's discuss about what are the different possibility for catastrophic losses in a case of conventional methodology of germplasm. There are four disadvantages like attack by the pest and insects or pathogens, climatic disorder, natural disaster, and economic causes. The conservation of germplasm can be done with the help of in situ and ex situ, as we discussed in earlier slide. Let's know what is the in situ preservation. In situ preservation, that means preservation of germplasm in their natural environment or habitat with the help of establishing biosphere or natural park like a natural sanctuary, and ex situ preservation in the form of seed or in vitro culture. So that is the main difference between in situ and ex situ. Let's discuss about in situ conservation. As we already discussed in earlier slide that in situ conservation is nothing but conservation with the help of natural environment or by establishing the biosphere. This in situ conservation, that is a high priority germplasm conservation program. The major limitation for this methodology is the risk of loss of germplasm due to environmental hazards, and the cost for maintenance of large number of genotypes is very high. So these are the two main disadvantages related to in situ conservation. Let's discuss about in situ conservation. As we already discussed during the classification of conservation, ex situ, this methodology is used for the preservation of germplasm which are obtained by cultivated and wild plant material. This genetic material is in the form of seeds or in vitro culture that can be preserved as a gene bank. Ex situ conservation has various disadvantages like some plants do not produce the fertile seed. Therefore, this methodology is not suitable for such a type of plant. During this methodology, some seed loses their viability. Seed destruction can be takes place by the pests and other options. Poor germination rate. This is only useful for the seed propagation plant, and this methodology requires high cost for processing. So these are the disadvantages of ex situ conservation. Let's discuss about advantages. They require small area, therefore amount of material is very small, and protection from environmental hazard. Therefore, environmental destruction can be limited in a case of excessive conservation. Let's discuss about in vitro method. In vitro method is applicable for shoots, meristems, and embryos. Advantages of in vitro methodology. Large quantity of material can be preserved in a small space. That is the major advantage of in vitro methodology. Let's discuss about advantages of in vitro methods. The large quantity of material can be preserved in small space. Germplasm preserve can be maintained in an environment which is free from pathogens. This germplasm can be protected from natural hazards. Large number of plants can be obtained with the help of germplasm stock whenever it is required for further experiment. Then, the obstacles for their transport through our national and international borders are minimum. These are the main vital advantages of in vitro methodology. Let's focus upon in vitro methodology. In this in vitro methodology, micropropagation, that means plant tissue culture, can be included. As you can see in this particular diagram, plant material it is used for in vitro initiation process. That means explants are selected. Explants are nothing but a part of plants which is used for the experiment, which are inoculated into a media which consists of different amount of growth hormone. Depend upon they will start the proliferation process and cell differentiation process that will lead to rooting and shooting process. After that, the generated plant it is used to transfer into a pot for acclimatization process, and then finally it is introduced into a field. This is nothing but a micropropagation method which is come under in vitro methodology of preservation. Then we will discuss about what are the different approaches in the case of in vitro conservation. The first approach is cryopreservation, second cold storage, and third one low pressure, low oxygen storage. Let's discuss about cryopreservation. Cryo name itself gives what is the basic principle of an preservation methodology. That is preservation at a very low temperature, that is a frozen state. Due to that frozen state, the selected plant material and tissue cultures are taken into a zero metabolism or non-dividing state due to a very less temperature. That is the main principle of cryopreservation. Cryopreservation can be takes place at a different temperature as you can see in this particular slide. The first one it is over the solid carbon dioxide at a minus 79 degrees Celsius. Low temperature deep freezer at a minus 80 degrees Celsius. In vapor phase nitrogen at a 150 degrees Celsius. In liquid nitrogen at a minus 196 degrees Celsius. So these are the temperature which is useful for the preservation of an germplasm. Let's discuss about what are the stages of cryopreservation. The first one, development of a sterile tissue. The sterilization step is very important to delete the different type of contaminants that can be hazardous for the further process. The second one, addition of a chiroprotectant and pre-treatment that will delete or that will decrease the risk of cell destruction. Then freezing, then storage, thawing, reculture, measurement of viability and plant regeneration. So these are the basic steps for cryopreservation technology. Let's see the example of cryopreservation. Let's start from shoots. Shoot tips are added into a pre-growth on a media in the presence of DMSO. DMSO it is a cryopreservant which is decrease the risk of cell destruction. Then there is an addition of an chiroprotectant in a culture media. After that, these shoot tapes are frozen in a liquid nitrogen, as you can see in this particular diagram. Then these ampoules are stored in liquid nitrogen, which is further used for thawing process, and then shoot tapes in a semi-solid media that will again converted into shoot tape culture that will use for regeneration of an shoot. That is the first methodology. In a case of second methodology, after addition of an chiroprotectant, that shoot tape, it is added in a liquid nitrogen that is plunged into a media at a 25 degrees Celsius. Again, this shoot tape in a semi-solid media further used for culturing technology, and then there is a generation of a shoot tape. So that is the technique for a cryopreservation. In the next slide, the same methodology it is shown in a diagrammatic form. That is cryoprotectant solution. Then further step is freezing and rebottling. Post culturing, that is culturing into a solid media. Plant regeneration. Then material and type of quality is going to be checked. Induction of an cold and desiccation tolerance, and again cryoprotection solution. So that is the simplest representation for the cryopreservation. In this slide, I gave you the reference for the YouTube in which a detailed procedure of cryopreservation is given for better understanding of principles and methodology of an cryopreservation. Let's discuss about application of germ storage. Germ storage it is used for maintenance of a stock culture for the several years, and it is useful for the experimental procedures. Then it is the ideal method for long-term conservation. In this methodology, the disease-free plant material can be frozen and it is used for propagation purpose whenever it is required. It is most commonly applicable for the relaxed citrant seed, which is maintained for the long period of time. It is also useful for conservation of somaclonal and gametoclonal variation. It is most commonly utilized for endangered species and pollen for enhancing the longevity. The rare germplasm developed with the help of somatic hybridization and other genetic manipulation processes. This methodology it is very suitable for the cold resistant mutant cell line, and it can be useful for generation of germ banks. So these are the applications of germ storage. Then let's discuss about limitation of germplasm storage. It is very expensive procedure because it requires the expensive equipment and trained person for the procedure. So today we discuss about germplasm conservation methodology, different classes, in situ, ex situ, in vivo methodology for the application of germ storage and limitation of germ storage.